Hello everyone, this is Mesam Ali and this is my progress report of my final semester of the master degree. So this is my progress report checklist which I reviewed from my respected professor Ong Kong Chai, and this is the selected courses in my final semester. I finished my all courses in previous semesters, so in this semester I only took seminar 4. I finished all scholarship compulsory subjects and all required courses to fill the criteria for graduation. This is the bench work which I learned in my this semester, that is bacterial culture for bacteria keratitis disease model and bacteriophage purification and culture. So this is the progress report title in this semester, like the name of my title of project for this semester is only antimicrobial effect up to 2 nanometer far-UVC on bacterial keratitis disease rat model. So this is the study design. This time we induce the BCRC number 10261 strain of bacteria to infect the cornea of the rat to develop the bacterial keratitis disease model. So this time we have 4 different groups. Each groups have around 6 rats. This is the no treatment group. This is the piperacillin and amikacin. These two are the drugs which is used against to kill the bacteria, and amikacin is used for severe cases. And this is the UVC for 3 seconds, 8 times a day. This is the treatment time point, 8 times a day. So the duration of this whole study is around 14 days. So we infected the cornea by using 35 gauge needle to make the suture of the cornea like this, and then we induce the bacteria by the pipette. Only 1 or 2 drops is enough for each rat eye. So later we collect the bacteria from the cornea onto cotton swab up to 14 days on different days, on several days, not every day. So this is the process of wound healing on cornea, how the bacteria is penetrated to the lower layers of the cornea. So this is the epithelium, stroma and epithelium. This is the general structure of the cornea. So at 0 hour the bacteria is not penetrated truly to the whole stroma. Later, after 24 hours, the bacteria will be penetrated to the stroma of the eye. So this is the picture we checked by a fluorescent strip after 24 hours. So we checked here like the bacteria is much more available. So this is the cornea scratch. We use the 35 gauge model, 35 gauge needle to suture to wound the cornea, and later we induce the bacteria. So this is the result after 22 hours is like that. After 96 hours is much more severe, like it's much more colorful compared with 22 hours, and later we sacrifice after day 14. So this is the collect bacteria after 24 hours for bacterial culture test. So when we induce the bacteria, so after 24 hours we collect the bacteria from each rat eye to check the bacteria still available on the rat eye or no. So we checked here in Suchi hospital from the bacteria lab. So they check like each group have too numerous to count. I mean that they have a high bacterial concentration on each group rat eyes. So this is a treatment started from day 1st. So this is the treatment results on day 3. So this is the control group. This is the piperacillin and amikacin. This is the drugs only. This is the UVC group, and this is the UVC and piperacillin and amikacin combined group. So I gave treatment to these rats for 14 days, every day from 8 o'clock at morning to 10 o'clock at night. So this is the result of different day. And from day 7 we started scratch cornea 1 time per day. So it means that it's also used sometime in a clinic when there is a severe case of the bacterial keratitis. So doctor also prescribed to the patient to use the cotton swab, and they have some special protocol that how you can use the cotton swab on the eye to remove the debris, because sometimes when the patient using the drugs, so sometimes the debris come out from the cornea but it's still attached to the cornea. So if we use the cotton swab, so it will also help you to take the debris out from the eye. So this is the treatment result on day 9, and this is the scratch we again did on every day, and this is the result on day 9. So this is the result of day 11. Here again you see like from after day by day, like the result, the morphology of the eye is look like very clear. So after day 11 giving treatments, we again collected the bacteria from different groups and we checked from the Suchi Hospital bacteria lab. So they again give us the result that here is not a viable bacteria available because of you are giving the treatment this or the colonies, but this is not the strain of bacteria which we use, this is some other bacteria. So this is the result of day 14. Here we also check the result of different groups. So now here we check the summary of each group effect of using the different drugs. Like here in this picture you check like piperacillin and amikacin eye drop have the strong effect, like all the eye on day 14 was clear compared with the day 3 or day 0. And this is the treatment effect of UVC therapy only from day 0 to day 14. And this is the effect of UVC light and piperacillin plus amikacin eye drop combined group, so it also have the good result. So we again collected the bacteria on day 14, so we did not find any kind of bacteria which we induced to the rat eyes. So this is the future studies. So future studies analysis is only to an experiment related to bacterial keratitis disease model or under process to check the effect of eye drops against bacterial infection to treated groups and UVC to 2 nanometer radiation groups. So there are some other experiment or under process that are HNE staining, PES staining and IHC or still under process. So ongoing research is also evaluate the different treatment method for bacterial keratitis. So we also, like this time we only use the eye drops and UVC radiation. We also have the pitch therapy for those type of bacteria which are multi-drug resistance. So this therapy offers advantages such as specificity, potential replication within bacterial population and the ability to overcome antibiotic resistance. Further research is needed to optimize pitch selection, dosing and delivery method, but it holds promise for treating multi-drug resistant bacteria infection like infectious keratitis. So we are doing this pitch therapy and other these experiments are under process. So we are doing this hopefully in near future. Thank you for all of you.